So here is what we're going to talk about. Brought to you by Curious Moranland. If you were to look at my tie, you'll see, and my wife got this for me, so a whole bunch of neurons. Let's stand up so you can see. All right, so look at this. This is a neuron, and over here is a neuron. And they're not touching, but they're communicating. Let's see if you can zoom in and see all those. So those little dots, those are a communication tool saying, hey, I've got information, I want to pass it on to you. And we're going to learn about neurons and something called a synapse and the communication tool, which is called a neurotransmitter. So let's start. And in this video, I'm going to pretty much cover this basic outline. This is going to be kind of a busy video, so definitely pay close attention. And you'll want to pause it at different points. So let's start. So if you look here, this is a neuron, and you notice the direction it's going to connect to this neuron. Now, there's a little gap there. Here's a neuron. There's a little gap. Here's a neuron. There's a gap. Each one of these little gaps is a synapse. All right, these, look at the different synapses I have labeled here. Individually, it'd be synapse. So what I want you to see the communication between neurons here, muscles, and glands. We always seem to forget about the glands. And if you look, you'll notice this is a multipolar neuron, and we could see unipolar connecting and still have a synapse. But let's add some labels. Let's remind ourselves we have the efferent or afferent. Now I need to add something to show the afferent in a second, but efferent is going to be a multipolar neuron that's ultimately going to connect to a muscle or a gland. Now, if it's neuron to neuron, we simply just say synapse, but ultimately it's going to get to its target. This is what we want, the ultimate information. If we're talking about, say, a motor response, we're going to actually see this neuron coming down here and causing these muscles to contract, or causing this gland to, say, release a substance or make a substance. So we can have a synapse, or we can have a neuromuscular junction, which I have spelled down here, or a neuroglandular junction, which I have spelled down here. That is the cell-to-cell communication going to an actual response. So let's add some more details to this PowerPoint here. So technically, this would be the effector. Those muscles are going to contract, the neuromuscular junction. That's really the synapse, that last neuron that actually is innervating, meaning having communication and contact and stimulation of these muscles. Or it could be like, say, this is the adrenal gland. Now, if you notice I added effector here, I would have to, there's a slight problem though. I need a different neuron. So let's imagine this effector or receptor. This might be a sensory neuron. This is a poorly drawn one, but if you notice, these are its dendrites. There's the cell body for a unipolar one, and this would be the axon terminal that would connect. So imagine the effector, and then this would be basically effector. This would be the effector, ultimately communicating with the muscle or with the gland. So let's dig in a little deeper to this whole synapse. And there's some terms and information you need to understand. So let's start here. The term ligand is it's a signal molecule, and cell-to-cell communication is a huge tool, and it's how your body physiology works. They don't have lips, they can't say, hey, do this, so they communicate through electrical stimulation or chemicals. So each one of these little ions here is going to pass through this membrane. So here's a membrane, the gate is closed, this signal here attaches and it causes a conformational change that opens this gate, this protein channel, and these ions are going to diffuse through. Facilitated diffusion, or usually it's active transport, because there's going to require some energy, and it's going against the concentration gradient. So, and then what happens is it dissociates, it breaks off. And so over here, just a quick baby bio review of exocytosis. See, these right here are vesicles. Now we're going to talk about what's inside those vesicles. Ignore the fact that these are little green dots and these are green dots. and I'll elaborate in the next slide here. So for now, imagine this being cell 1 and this being cell 2. So here's cell 1, right, that's 1, and then this is 2. This would be these would be cells 1, and then this would be 2. Now later I'm going to use a different term for that, but really what happens is the ligand, whether it's nervous system or endocrine, they're both your means of control, and they're signaling. So what happens? Cell 1 sends a neurotransmitter across this little space. We'll learn that's called a synaptic cleft, and it's going to interact with this cell, and that cell is going to do something. So look at my little notes here. Cell 1 releases the ligand. The ligand travels to cell 2. Cell 2 accepts that ligand, and then cell 2 responds by passing its message on. Later on, we're talking about, say, passing that action potential on to the next one, which is really what a synapse is. In this case, there's going to be some distance. The neurotransmitter, in this case, it's a ligand. It might be some type of hormone. Instead of being a neurotransmitter, it's going to be a hormone. It gets into the bloodstream and travels a longer distance to the targeted cell. So let's look in here. This is just looking at, here's cell 1. The ligand is actually a hormone, and it's going to travel the distance, and it basically affects, say, a skeletal muscle or a gland, or that's pretty much, or even nervous tissue. All right, so take a quick look at this. And so here's our synapse again, and let's zoom in, and notice some labeling that I've added here. So this is your synapse. This is cell 1, which is the end of one neuron. That's called the pre-synaptic terminal. And then there's the synaptic membrane. So the stuff that's in here, those are neurotransmitters. See, these neurotransmitters over here, they are going to cause something to happen to the next cell. So this is cell 1. This would be this cell 2 actually is over here. So that's cell 2. Cell 1 is here. The presynaptic membrane means the cell membrane, the plasma membrane of the axon. The end of the axon is called the axon terminal. Now inside each one of these is, imagine a whole bunch of neurotransmitters. So let's go to the next slide. And what the cholinergic synapse is what we're going to do. We're going to say the action potential arrives. The ACH is a neurotransmitter, so it's going to be released and diffuse across that cleft, and that's going to trigger a depolarization of the post-synaptic membrane. So this post-synaptic membrane is actually cell 2. And then what happens is you're going to recycle this. So let's go ahead and go talk about what's happening here. Now, each one of these is a neurotransmitter. Each one of these little circles. At this point, the action potential arrives. Notice these are sodium gates. They're closed. They're closed right now. They will open. What's going to trigger them to open? The neurotransmitters. So here's this is cell 2. This is cell 1. Let's watch what happens. All right. This is ACH, acetylcholine, and this molecule right here, right there, is acetylcholine esterase. This is going to break down the neurotransmitter when this whole synapse is done. So the action potential is coming down in this direction. There's no sodium gates here. Remember, the dendrite is what we call a stimulus receptive membrane, and there's a high concentration of sodium gates. There's no sodium gates. So we talked about sodium and potassium and depolarizing and hyperpolarizing, but there's a new ion that kicks in, and that's calcium. So here's calcium. Calcium goes in these gates because it's a reaction to the action potential that's been coming down this direction. So these gates, another voltage gates, they open and trigger the synaptic vesicles, that's these, all right, to go through exocytosis. That means they're going to come in, they're going to fuse, and here's the neurotransmitters. At this point, the gates are still closed, all right? And the neurotransmitters, when these land onto here, they're going to cause the gates to open. Now, all this sodium is flooding in. What is it when you have a rapid change in the membrane potential? You're depolarizing. So now all these are now plus on the inside because these sodium ions are going in. Now, if this is a strong enough signal, enough neurotransmitters are going to be released, all right? They are going to depolarize the postsynaptic membrane. And this is depolarization. This is those sodium ions going in. And it's going to work its way down the axon, meaning it's going to hub at the axon helix from the dendrite. So this actually is the dendrite right here. This is still positive and negative on the over here, but as it works down, this is going to flip. So then this will become negative and positive on the other side. All right, so this is really what's going on with the synapse. The last thing is you're going to move it down. So if you might have noticed, this is now positive on the inside, positive on the inside. If you go back to the previous one, see I had it positive, negative now, and this look a little color change there, make it blue, and so now it's there. You'll notice the neurotransmitters, they've detached. So these sodium channels are closed, and now that enzyme is going to break this down. So I want to elaborate on this with the next couple slides. So let's look at this. Pause the video and go ahead and read through here. And here's just some other examples, just zooming in. Here's the neurotransmitters coming in. They attach, they serve as a ligand, which causes that gate. Here it is opening, and that allows for sodium ions to pass through. And this is just here's a zoom in. Here's the sodium ions going in. When the ligand detaches, no more sodium can pass through because this gate has closed. So you'll see the ions close, ions close. So now let's look at this picture. So the action potential is moving down. So step one. So this is just kind of a review. Step one, the action potential moves down, causes the calcium to diffuse in. This triggers these synaptic vesicles, which are holding a neurotransmitter. We're actually talking about acetylcholine is the neurotransmitter. So neurotransmitters are they're these secretory vessels, actually synaptic vesicles. They're going to come, they're going to fuse. So step two is move down to, they're activated. Step three, they're going to diffuse and travel across the synaptic cleft. This is the synaptic cleft. Again, this is the pre-synaptic, this is the post-synaptic. This is an axon, this is a dendrite. So now you have this rapid depolarization, meaning the sodium ions are flushing through. When this depolarization, and you notice the action potential is going to wave. Now they're showing two, it's not like it can go in two directions. This is going to run down, hub at the axon helix and run down the axon, and the action potential is now being passed on. That's really what a synapse is a communication, the action potential of one neuron passing to another neuron, or a neuromuscular junction, or a neuroglandular junction. So let's dig in here. So now, step 6, most of my students understand this. This is how we do reuptake. So this ion right here, this neurotransmitter, sorry, it's used. It's done its thing. It's going to detach, and you have this enzyme, which is actually on the plasma membrane of the postsynaptic neuron. It's going to break it down. The choline goes back in, as you see it going back in. The acetic acid is going to go to the interstitial fluid, and if this is in the brain, this would be cerebral spinal fluid if you're talking about neurons. All right, so this is a way of recycling. Now the acetic acid you get through consumption, the choline you recycle back. This is something that you would actually have to uptake later, but they're not showing the acetic acid getting in here. But now once the choline gets in, the acetic acid that's in the cytoplasm of the neuron will recombine them. That's recombining. So this is a way of recycling. You get new acetic acid from the cytoplasm. The choline leaves in the form of acetylcholine. It's broken down and re-enters. This is called reuptake. So I'm going to come over here, and basically, instead of just talking through this, I'm just going to point out to my students, at this point you should pause the video. or look at the PowerPoint that has a slide and read through here. And I would do this. If you're studying, I would write out 1 through 9 and write a written description of what's going on here, and then check your answers with what I've posted here. All right, so that's pretty much a synapse from the start to finish, and the molecules that are used. So I'm going to do another video on neurotransmitters, and we'll elaborate on this reuptake and how these can stimulate an action potential or prevent an action potential, because we're going to talk about excitatory and inhibitory. So, all right.